The time saving basically is another way to measure the treatment effect. So traditionally, the treatment effect is measured by looking at a primary endpoint, for example, CDR sandbox, and then compares the mean change from baseline at the last study visit point. Traditionally, it's 18 months. This is a very good way to conduct clinical trial, but a difference in this primary endpoint is hard to interpret. So another way to try to make the interpretation of the treatment effect easier is to convert this absolute difference between the primary endpoint, like CDR sandbox, to a percentage reduction, which helps, but still is not intuitive. For example, lecanemab can reduce the disease progression by 27% after 18 months of treatment. That's a reasonable treatment effect, but what does the 27% really mean? To overcome this issue, people introduce the time-saving effect. Basically, it's another way to transform the percentage reduction to a time scale. In lecanemab, 27% slowing in the disease progression was converted to a 5.3 months time saving. And the method people use typically is, we call it backward projection to placebo decline method, which means you backward project the treatment group decline at 18 months to the placebo disease progression trajectory until at a time point they are equal. And then the time difference between the matched time point at the placebo decline versus the 18 months is the time saving. So this is the method people use, but this method has some issues. Just like any method, when a new method is proposed at the beginning, there are always some areas you can improve on. So we are what we are trying to do is to improve this method, and also to provide some reasonable interpretation of what you can get from this method. Well, the first thing we want to point out is that based on the published figures, the published papers, it's very easy to misinterpret the results from this method. For example, you see publication talking about a fixed absolute difference can be interpreted as an increased time saving over time. And the first thing we point out is in those methods or in those examples, there is some underlying strong assumption making those concepts appealing, but they are not realistic. The major assumption is that in those demonstration examples, the placebo actually demonstrate a very good treatment effect. The placebo basically lead slowing down in the disease progression over time. So that's a major issue that makes the time-saving effect more appealing than it is supposed to be. So we point this out and we demonstrate using example to show if you only have a fixed delta, fixed absolute difference between treatment and placebo, it's more likely that will lead to a fixed time saving or lead to a decreased time saving because the placebo decline will get worse. There are some other issues in this existing method. For example, when you project the treatment at 18 months backward to placebo at, for example, using lecanemab to 12.7 months. When you project backwards, it basically matching the different time points that lead to a kind of a disruption of the randomization equilibrium. And also, the existing method really doesn't take into account the delta. The delta is the absolute difference between treatment and placebo. Because it doesn't use this delta that lead to some time-saving estimation, which does not reflect the difference in the observed delta. Well, like I said earlier, every approach, when it was first proposed, there are ways you can improve. So what we are trying to improve upon the existing method, basically is trying to overcome the disruption of the randomization equilibrium, because this is a very serious issue in these clinical trials. As you probably observed, in almost all the Alzheimer clinical trials, the treated arm typically has a larger dropout than the placebo arm. So we want to avoid this disruption of randomization equilibrium, because that basically invalidates the statistical inference. And we want to utilize the observed difference in the trial between treatment and placebo. So based on this, we just come up with two methods which we believe statistically or clinically are improvement based on the existing method. One method, we call it backward projection to the treatment decline method. The other method, we call it the additional time needed to reach placebo decline method. Both methods utilize less assumptions and we think improvement based on the existing method. But the most important thing we want to point out is it doesn't matter how you interpret the data. Either you use absolute difference, or you use a percentage reduction, or you use a time saving. They all are derived from the same data. If you think 27% reduction in disease progression is not going to change or stop the disease progression, then a 5.3 months time saving is not going to do that neither. So it's the same data, different metrics. We should interpret the results. Well, I think the most important aspect of this is it really makes it much easier to communicate with every parties involved in the Alzheimer field. It makes it very appealing and intuitive to talk about the treatment effect. So I think that is the biggest advantage. Other than that, I think all the other informations, again, like I said, they come from the same data set. So I don't think it will fundamentally change any aspect of the clinical trial.